Our next speaker is also from Pittsburgh, and he is well known because he works with spermatogonia stem cells, but one of his passions is also how to protect fertility in patients. So he has a massive program of collection of and cryopreserved testes and ovaries, and also developing into the clinics. However, today he will be not talking about that directly. He will be talking about Sertoli cells. And on the top of that is my neighbor, and I always bother him asking things about NIH. So, Kyle.
Well, thank you, Miguel, and I'd also like to thank Yoel and the other organizers for, first of all, a great conference, but also the opportunity to tell you about one of the projects that's going on in the lab related to Sertoli cell gene therapy for male infertility. So, 1% of men in the global population have a condition called azoospermia. That condition means that there are 0 sperm in the ejaculate, and of course, it's impossible to have a biologically related child if you can't produce any sperm at all. Just as a side note, 1% of women in the general population have the condition premature ovarian insufficiency, which means no eggs. So men and women are equally impacted by this kind of a diagnosis. Well, in the United States, those numbers translate to 650,000 men between the ages of 20 and 50, which you might consider to be the reproductive years, and that translates to 15 million people worldwide. Now, half of infertility is believed to be genetic in nature. Well, what I should say is half of infertility is unexplained. Once we check off all of the likely causes and we don't find a cause, it becomes unexplained, and we think that most of that is due to genetic mutations. And today, with affordable whole genome sequencing, it's possible for us to not only discover the genes that might be the cause of the infertility, but also to use that information to develop diagnostic screens and potentially to develop targeted gene therapies. So the question is, if we were going to use gene therapies to treat male infertility, what are the medical, ethical, legal, and social implications of deploying those technologies in the clinic? And as a sub-point here, and this is really what I'm going to spend a lot of time on today, is what is the risk that the genetic change that you make in the testis might get passed to offspring? Because we don't know all we need to know about that technology yet, and we don't know what the implications might be for the unborn offspring. So even if we can use the technology to treat a man's infertility, if it has a negative impact on its offspring, that would be bad. And because of that uncertainty, and because this topic is widely debated around the world today, it's actually illegal to do this thing called germline gene therapy or germline gene editing in the United States if there's a possibility that the genetic change would be inherited by offspring. By the way, that's not universally true. There are other countries in the world where there may be a path forward to do germline gene editing, and if we could, it may be an option not only to treat infertility, but also many congenital diseases that have terrible impacts on the quality of life in patients. Well, to understand the issue of germline gene editing and how we might be able to deploy a gene therapy for infertility and avoid the risk of transmission to the next generation, I'll teach you a little bit about the process of spermatogenesis. So spermatogenesis is a stem cell-based system. Unlike the pluripotent stem cells that you've heard about today already that can produce all the different cell types in the body, this stem cell knows only how to make sperm. And like all stem cells, it's defined by its ability to self-renew and maintain the stem cell pool. And these self-renewing divisions are balanced with differentiating divisions that give rise to the spermatogenic lineage. And the terminal cell in that lineage is a spermatozoa that has a head that carries the haploid genome that dad will contribute at the time of fertilization, and a tail so that it can swim through the female reproductive tract and fertilize the egg. Now, all of this process of spermatogenesis happens in the seminiferous tubules of the testis. The testis is made up of a bunch of tubules that look like spaghetti. Sperm are made inside these tubules, and then they flow out to the reedy testis, and then via the efferent ducts to the head of the epididymis, and eventually come out in the ejaculate. If you take a cross-section through one of the seminiferous tubules, the seminiferous epithelium looks like this. And on the next slide, we'll zoom in to look at this in more detail, where the spermatogenic cells, or the germ line, is indicated by the blue colors, and the spermatogonia, the stem cells, are located down here on the basement membrane, and as they differentiate, they move further away from the basement membrane, differentiate into sperm that are eventually dumped into the lumen of the seminiferous tubules. Now, it's reasonable to suppose that some genetic mutations that cause infertility will be mutations that cause a dysfunction in germ cells, and therefore to fix that mutation, you would have to do gene therapy in the germ cells and increase the likelihood that the genetic change would be passed to offspring. But what's equally likely is that a mutation that causes infertility might occur in one of these other cell types that are present in the testis and that are necessary to support spermatogenesis. And one of those cell types that's shown in the salmon color here is the Sertoli cell, which is an amazing cell that we call the nurse cell of the testis. And that Sertoli cell supports every stage of spermatogenic lineage development, all the way from the stem cell to the terminally differentiated sperm. So the question is, are there mutations that cause male infertility, that cause defects in testicular somatic cells? Today we'll focus on the Sertoli cell. And could we repair those cells without having the risk of passing the genetic modification onto offspring? Well, to answer that question, we obtained a mouse model called SCARCO. That stands for Sertoli cell androgen receptor knockout mouse. Androgen receptor is the receptor for testosterone, and testosterone is absolutely required to support spermatogenesis. So if you knock out the androgen receptor, testis can't respond to testosterone. And the result is, if you look at the testes of these animals, they're much smaller than wild-type animals. If we breed them, they don't produce any offspring compared to the controls that produce lots of offspring. And if you look at the histology of the control animals, you can see that it looks like the cartoon that I showed you, with a wide open lumen in the middle, and I think you can imagine the wispy tails of sperm here in the middle. In contrast, the SCARCO mice have smaller tubules. There's lots of round germ cells in here, including the stem cells, but there's no lumen and there's no sperm. So Chachnan Domkemchan, when she was a PhD student in the lab, asked the question, would it be possible to use a gene therapy to correct the androgen receptor mutation in these animals, or actually add back a functional copy of the androgen receptor? And she decided to do that by using an adenovirus. The reason she used an adenovirus, first of all, is it doesn't integrate in the genome. So that means even if this virus got into a germ cell, if it doesn't integrate in the genome, it's unlikely to be passed to progeny. The other characteristics of an adenovirus that we knew from 20 years ago in my own failed experiments is that they very effectively transduce Sertoli cells, and they completely fail to infect germ cells. Of course, 20 years ago, I was trying to infect the germ cells, so that was a failed experiment. But for this application, it's exactly what we're looking for. So Chachanan developed two viral vectors. One contains the human androgen receptor in red. The other one is the empty vector control. And she injected those viruses into the seminiferous tubules of the testis with the hope that they could add the corrective gene to Sertoli cells. The result of her experiment is shown here when we collected the testes several months later. The animals that got the control vector, so no corrective gene, still have the same phenotype, lots of round germ cells, but no lumen and no sperm. But the animals that got the corrective gene, you can see there's a dramatic change in phenotype where the corrected spermatogenesis has multiple layers of germ cells, a wide open lumen, and sperm in the lumen of the seminiferous tubule. So the next question was, are those sperm functionally competent to fertilize mouse eggs? And the answer is yes, we could fertilize by intracytoplasmic sperm injection. That resulted in the production of 2-cell embryos, 4-cell, 8-cell, and etc. Now in a mouse, we typically transfer embryos at a 2-cell stage, so we did that. We transferred these embryos into a surrogate female, and she was able to have babies. And then those babies could grow up to adulthood and have babies of their own. So it appears that this adenovirus-mediated Sertoli cell gene therapy could be used to restore spermatogenesis. The next question was, is it is there a risk of passing that genetic modification on to offspring? And the answer appears to be no, because when we when we bred the virus-treated animals, they produced lots of babies, and out of over 400 babies, none of them had the green fluorescent protein, which was also included in the therapeutic vector. Well, this is kind of the contrived situation. We knocked out a gene, we knocked it out specifically in Sertoli cells, which isn't the way that it normally would work. But just to summarize this part of the talk, we think that Sertoli cell gene therapy is technically feasible today, at least in mice, simply by injecting in vivo a replacement copy of the defective gene. It may be possible to do Sertoli cell gene therapy without having germline modification that leads to transmission to the next generation. We did observe that adenovirus had some toxicity to the testis, and so that's something that we'd like to avoid. And also, we still need to discover the genes. We know actually a vanishingly small number of genes that are associated with male or female infertility, so there's a lot of discovery work that still needs to happen. So to begin tackling that problem, I want to introduce our team of multidisciplinary and also multi-institutional investigators that's funded by an NIH program project grant to discover the genetic causes of infertility. The team members are John Scamenti from Cornell, Don Conrad from Oregon Health and Science University, and on the bottom, Alex Yatsenko from here at Magee. Oops. I also want to introduce all the graduate students that participated in this project at various different levels, from recruiting the patients and discovering the mutations that actual patients have, developing mouse models to prove that the mutations are associated with infertility, and then developing gene therapies either in testicular somatic cells or testicular germ cells. So basically the way this pipeline works is that we're recruiting patients with that most severe infertility diagnosis called non-obstructive azoospermia. We recruit patients and collect a sample of blood, and then we do whole genome or whole exome sequencing to see whether there are any genetic variants in that patient's genome that could be related to their infertility. Of course, when you do this, you discover hundreds of mutations in every patient because we're all walking bags of mutations. It's what makes us different from each other. But we can tease through that using bioinformatics and find some most likely candidates. And when we find a likely candidate, we use CRISPR-Cas9 gene editing to introduce the human mutation into the orthologous space of the mouse genome. So that assumes that part of the mouse genome is conserved from human to mouse. And then we evaluate the fertility of the mouse. If this mouse is infertile, then it proves that the mutation that we found in the human patient is most likely the infertility-causing mutation. But now also we have a mouse model of a human patient that we can use to develop a gene therapy. So we've been recruiting our patients both at the Oregon Health and Science University with Don Conrad and also here at Magee with Alex Yatsenko. And we have done whole genome sequencing or whole exome sequencing on thousands of patients already. And here's a list of just a few of those mutations. To see what happens when I click again. Right. And so the ones on the top of this table are mutations in genes that are expressed by Sertoli cells. So we think that it's specifically a Sertoli cell dysfunction and not a germ cell dysfunction. And you can see here that there's two genes, CLCN2 and MAP7, where mutations were covered in Don's cohort at OHSU. Alex here at MWI also found mutations in the same gene, and so that's one way to validate a gene. If two different studies identify mutations in the same gene, it is suggestive that it might be involved in infertility. Now, before I go on, I also want to point out that we're not selective just for somatic cell genes. We also have mutations in germ cell genes, and we are doing germline gene therapy in the lab. And so topaz1 is another mutant. By the way, we're in the process of making mutant mice in with all of these genetic mutations. But I'll use this slide just to summarize data that I don't have time to tell you. And one is that we have created this mutation in a mouse. We've done the fertility testing, and this mouse is infertile, so it validates the mutation that we identified. Similar, this topaz1 mutation, we produced mouse models. Those mice are infertile. This CLCN1 mouse is not infertile, and then we have MAP7 mutants that are still in development, so fertility testing is coming soon. So the CLCN model that I told you is infertile, now that I've shown you a cartoon of what testis seminiferous tubules are supposed to look like, hopefully you'll appreciate that wild-type mice have a seminiferous epithelium that looks like the cartoon, but the mutant mice have seminiferous tubules that are almost completely devoid of germ cells in the seminiferous tubules. And this is my last slide to show you that in addition to having produced the mouse, Georgia is developing the tools to do Sertoli cell-specific gene therapy. This time she's using adeno-associated virus, which seems to be less toxic to the testis than the adenovirus that Chachanan used. And what she's doing here is showing you the germ cells in white. So these are all the germ cells in the seminiferous tubules in white. She's also showing you the Sertoli cells, which are indicated by the red color. And what we find is that the virus that carries GFP overlaps only with the SOX9 positive Sertoli cells, and we don't see any overlap with germ cells. So that suggests that we should be able to do Sertoli cell-specific gene therapy with this vector without the risk of germline integration or transmission. So this is my last slide. I tried to acknowledge both our funding and also the people who did the work as I went along. And where I'll leave you is that I think now that Georgia has both the mouse and also the tools for genetically correcting the mouse, and of course that tells you what she'll be doing next. So with that, I thank you for your attention.